In this video, we're going to learn about neutrophil extravasation, also called diapedesis, and NETosis, which helps explain the phenomenon of inflammation. First, let's talk a little bit about neutrophils. They are a type of innate immune cell from the myeloid lineage. They are also granulocytes, so in the same family as basophils and eosinophils. Granulocytes are named that way because they have granules that contain proteins which help fight infections or mount an immune response. Finally, neutrophils circulate freely in the bloodstream, which means that they are very motile and they don't typically like to be attached to surfaces. However, during inflammation, neutrophils will migrate to the site of injury or infection, which requires them to adhere to the endothelial lining of blood vessels. So how does this happen? The process of diapedesis begins with chemokine signaling, which tells the neutrophil that there is a nearby injury or infection. Chemokines are a small family of cytokines that are important throughout this entire process. First, we have rolling. Selectins, for example L-selectin, P-selectin, and E-selectin, work to slow the neutrophil along the vascular trail. Selectins bind to specific sugars on the endothelium, for example PSGL1, which is P-selectin glycoprotein ligand 1. Selectins is actually a combination of the words selected and lectins, with lectins meaning sugars, so that's an easy way to remember what selectins are. Next we have activation. Integrins, for example LFA1, on the neutrophil switch from an inactive state to an active state, and this is mediated by chemokines. Integrins are a family of glycoproteins, and their job is to help cells adhere to other cells or to the extracellular matrix, or ECM. Then we have arrest or adhesion. Integrins on the surface of the neutrophil bind to ligands on the surface of the endothelium, for example ICAM1, which is intracellular adhesion molecule 1, and this allows the cell to firmly attach. Ligands like ICAM1 are upregulated specifically during inflammation, so this helps explain why the neutrophil is not always attaching to the endothelium. Finally, we have transendothelial migration. The neutrophil squeezes between endothelial cells of the blood vessel, which allows it to exit the blood vessel and enter the extravascular space. Once the neutrophil has exited the blood vessel, it will not re-enter the bloodstream and recirculate. Chemokines, for example CXCL1, will continue to guide it to the site of tissue damage or infection. Neutrophils respond to chemokines when the chemokine binds to its receptor on the neutrophil. Once the neutrophil reaches the threat, it will undergo a unique type of cell death called NETosis. Neutrophils are actually the only cells that can do this, and this explains why the neutrophil will not re-enter the blood vessel, because its only job is to take care of the threat, and that causes it to die in the process. During NETosis, the nuclear membrane of the neutrophil breaks down and the chromatin decondenses. Reactive oxygen species, granules, and chromatin stick together, and the cell breaks open to release nets, or neutrophil extracellular traps, which neutralize the threat. You can think of nets kind of like webs made of chromatin and other proteins that catch and kill the bacteria. The neutrophil granules contain really potent antimicrobial molecules that are able to kill the bacteria. Thank you so much for watching, and I hope you found this video helpful.